Hello and welcome to episode 3 of CCLG's Research Talks for Parents and Public. I'm Ellie Wilkinson and my job is sharing our research with our supporters. Let's start with housekeeping. So we'll be holding a Q&A session at the end of the talk, so please submit your questions whenever you think of one using the Q&A button at the bottom of the screen. If you'd like to use the chat function, make sure you've set the to drop down to everyone or people may not see your message. So this webinar is being recorded and we will send out the recording in the next few days. So this month's talk is called How Can We Improve Quality of Life for Children with Lymphoma and is being led by Professor Suzanne Turner. Suzanne Turner is the Professor of Childhood Cancer Biology at the University of Cambridge, whose research lab is working to improve childhood cancer outcomes internationally. She has a particular interest in the causes of childhood lymphoma and in finding biomarkers that support kinder treatments. So without further ado, I'm going to hand over to Suzanne.
Thanks very much, Ellie, and good evening, everybody. I'm just going to share my PowerPoint with you. So hopefully you can all see that now. Is that good, Ellie?
Looks good.
Yeah. Wonderful. So really it's a pleasure to be here and to be able to present some of the work that we've been doing here in Cambridge in my lab. And as Ellie said, my name's Suzanne Turner. I'm a Professor of Childhood Cancer Biology here at the University of Cambridge. And work in my lab really focuses on trying to understand why children get cancer, and by knowing a little bit more about that, what we can do to treat it better and maybe one day to even prevent it. And I'm going to focus today on lymphoma, as Ellie said, and the work that we've been doing there. And I'm going to start with a little bit of background. Of course, many of you will know that, of course, children are not mini adults. And so we shouldn't expect their cancers to be anything like what we see in adults. And if nothing else, we can see that very clearly here. So you can see that adults get very different cancers from what children do, and even young adults and adolescents. So most adults get breast and prostate cancer, followed by lung and colorectal cancer, whereas children tend to get leukemia, brain tumors, tumors involving the bone, and then this category here called lymphoma, which is a cancer of the immune system. And as we move into the young adults, again, we see a shift, but still we see a majority of these young adults and adolescents getting lymphoma, carcinomas, and germ cell tumors. So really a different spectrum of tumors in children to what we see in adults. And the cancers that develop from the immune system develop from very specific types of cells within the immune system. So we all know the immune system protects us from infection, but maybe what you don't know is that the immune system is made up of many, many different types of cells, all which have very distinct roles. And there are two types of cells in particular. There's a group of cells called T cells, and there's a group of cells called B cells. And I like to say these are a bit more like the targeted missiles of our immune system. So these are the cells which actively go out and seek infections. So they go out and look for all those viruses and parasites and things that shouldn't be in our body. Whereas other components of our immune system are maybe a bit more like sort of a carpet bombing approach. They just go around setting off little bombs whenever they see something that looks a bit abnormal, but not in a very educated manner. So these are the trained, educated cells of our immune systems. And these are the cells that develop into lymphoma. And so there are two broad categories of lymphoma, particularly when we talk about this type of non-Hodgkin lymphoma here. So non-Hodgkin lymphoma as opposed to Hodgkin lymphoma. And because they arise from T cells and B cells, we call them B-cell lymphomas or T-cell lymphomas. So quite simple, really. And when we look at the type of lymphomas that we see, particularly here, the example given is in Europe, the large majority of lymphoma, non-Hodgkin lymphoma that we see in children are sporadic Burkitt lymphomas. And Burkitt lymphomas arise from those B cells in our immune systems. And it's called sporadic in the Western world when this type of lymphoma occurs in, for example, sub-Saharan Africa, where malaria is endemic. It's then called endemic Burkitt lymphoma. And there it has a different mechanism to what we see here. The other type of lymphoma we see in children is another type of B-cell lymphoma that we call diffuse large B-cell lymphoma. And then there's the types of lymphomas that arise from the T cells of the immune system. And the predominant one we see there in children is this one here called anaplastic large cell lymphoma. And then there's this category called lymphoblastic lymphoma, which can be B or T cell. So very distinct types of lymphoma that we see in children. And bear in mind that there are probably about 80 different types of lymphoma that we see in adults. So it's a very different range of cancers that we see in children. Now, the good news is that most children diagnosed with lymphoma do very well with treatment. And so this is an example here. This is for B-cell lymphoma. This was the most recent worldwide trial that was ran recruiting children with B-cell lymphoma. And you can see here the number of children that survive is very good. So this is 5 years of follow-up. And you can see out of 100 children, 94 of those children survive for at least for 4 years. And of course, we hope that that will continue and likely will. There's another way of looking at these sorts of data. So if we also look at event-free survival, so this is the time between treatment starting and an event happening, which can be the cancer coming back, so what we would call a relapse, or the cancer getting worse, for example. And the number of children that have an event is basically only 7 out of every 100 children. So this type of cancer, this B-cell lymphoma in general, has a very good outcome. But of course, all of those of you who are parents of children with cancer will know that we're treating children with some very toxic, quite nasty drugs, which make them really sick. And of course, the reason for that is because most of the drugs that we use still today in the clinics are drugs which aren't specific for the cancer cells. So essentially, what we do is we give a drug to the children which kills everything that moves within them. So the regeneration of the intestinal tract, making new red blood cells, all these types of things that our bodies are constantly remaking are the cells which are also killed by chemotherapy. And this leads to the side effects that we see in children undergoing chemotherapy. So the tiredness, they don't have enough red blood cells carrying oxygen around their bodies anymore, feeling sick because we're getting rid of the lining of the intestines and the gastrointestinal tract. And so this is where the problem lies, particularly with cancers like this, where we have good outcomes. And so what's wrong with existing therapies is that they're not specific enough. And actually, it's really easy to kill cancer cells. I have students in my lab who sometimes not on purpose find it really easy to kill cancer cells. And that's because when we've finished working with them in the lab, we just put them in a bucket of bleach and the bleach will kill them. But of course, what we can't do is put children in a bucket of bleach. It's going to do a lot of damage and not just kill their cancer cells. So the difficulty is how do we kill cancer cells and spare the healthy cells in the body? Because ultimately the cancer cells are hijacking, they're using all the same mechanisms that our healthy cells do. And so I'm going to talk a little bit about that, but illustrate what we've been doing in the lab by focusing on 2 distinct types of lymphoma that we see in children. First, I'm going to talk about the T-cell lymphoma, anaplastic large cell lymphoma. And then I'm going to move on to a type of B-cell lymphoma, Burkitt lymphoma or B-cell non-Hodgkin lymphoma. So ALCL, or anaplastic large cell lymphoma, as I said, develops from the T cells of our immune systems. And it comes in one of many different flavors. So generally, it's called systemic ALCL, and that just means it can be anywhere in the body. It's systemic. But further on from that, we can break it down into 2 different types. There's ALK-positive ALCL. So this is a protein called anaplastic lymphoma kinase, ALK or ALK. And that's what we see largely in children. So most children that develop ALCL will have this systemic ALK-positive form. There's a systemic ALK-negative form, which is largely seen in an older population, extremely rare in children, and I'm going to touch on that as well. There's a skin-based form called cutaneous ALCL, and this new form that's associated with breast implants, which hopefully we'll never see in children. And of course, I'm not going to touch on that today, but we are also working on that in the lab. So what is ALK? So ALK, as I said, it's a protein in the cells of our bodies. And it's normally expressed in the nervous system. And it's this big old protein, and this is a cartoon here, try to illustrate what it is, but it sits in the membrane of the cell. So if we think of cells as being like a circle, what ALK does is it normally sits sticking out of the cell. And what it does is it looks for certain types of signals from its environment. And when those signals, those other proteins or receptors or other little molecules bind, what it does is it forms a dimer, so two portions of ALK join together. And then what that does is it allows it to become active and send a signal to the cell to tell it to divide and grow. But it needs this signal, it needs this to bind to enable it to be active. Now, what happens in ALCL, in the cancer cells, is there's a genetic event. And that genetic event takes this little intracellular part, the bit that's inside the cell, and it binds it to a new part of a different protein. And what this serves to do is means that it's not sticking out the cell anymore. Now it's completely inside the cell. And on top of that, it no longer needs this little signal to be active. So it's independently active and switched on in the cancer cells. And so this is what's driving growth of those cancer cells, the ALCL. And for ALCL, we do have some biomarkers. And by biomarkers, I mean things that we can measure from the patient, from the child, which might tell us a little bit about their particular disease. So for ALCL, what we know is that some children with ALK-positive ALCL produce antibodies to ALK. So it's almost as if their bodies are trying to get rid of the cancer cells. They recognize that this new protein shouldn't be there, but they're not overly successful in doing that because the cancer's still there. And the other thing we can look for is something we call disseminated disease. So has the disease spread into the blood or into the bone marrow? And we can look for that by taking blood samples or bone marrow samples and looking for genes, looking for that ALK gene, that new encoded protein that are sort of floating around in the blood and the bone marrow. And what we know from studies that have been done is that children that produce high levels of those antibodies and they don't have the disseminated disease, so it hasn't spread in the blood and the bone marrow, these are the children that they do really well with therapy. So you can see here, the survival rate is reaching 100%. And this is 12 years of follow-up. So this is a very long time after diagnosis. These children all do extremely well. And then we have a group of children who don't do as well on therapy. And these are the children which either don't produce antibodies or have very low levels and has disease that's maybe spread around the body. And so for these children, the outcome is still fairly good, but there are more events for these children and they do relapse and they don't seem to do quite as well with chemotherapy as we would like. And so what we're able to do now, what we hope to be able to do in the future, is to take this group of children here, what we call the high risk group, and be able to adapt their therapy so that we get this line here up here with the children that have the ALK antibodies. And so that's work that's in progress. And knowing about that ALK protein means that we can also generate a therapy that's very specific to those cancer cells. So I told you that they need this activator, they need a signal to activate the protein, and that when it becomes active in the cancer cells, it doesn't need that activator anymore. But there's something else that this protein needs to be active, and that's energy. And energy comes in the form of something we called ATP, and that's essentially the key. They don't only need the activator, but they need the energy. And so if we can block that energy, if we can prevent the energy getting to the protein, we can stop it from working and stop it from telling the cancer cells to divide. And so drugs have been developed by companies which enable us to do that. So this is looking at a sort of a cartoon of what we think the protein looks like. And it has this little pocket here. You can see it a bit more up close here, this little pocket here. And this is normally where that ATP energy sits. It sits in that pocket and it gives that protein all the energy it needs to tell the cells to drive, to grow, to proliferate. And we now have drugs such as a drug called brigatinib, or you may have heard of crizotinib or lorlatinib. There are different forms of this drug which sit in that energy pocket. They stop the energy getting in and stop that from working. And this kills the cancer cells. So we now have a drug which is called a targeted agent because it's very specific to the cancer cells. So it has very few side effects, still has some, but very few side effects that we can use to treat the children. Now, there are trials which are starting now to get these drugs to children. Unfortunately, there has been some reluctance from drug companies to trial these drugs in children. So much of the work that's been done to date with this drug has been with adults that have ALK-positive lung cancer. But we are starting to be able to access these drugs now. But I want to put it another way for you. Hopefully that was all very understandable, but it's much like having a key to a car. So that protein, like a car, to activate the engine, it needs a key. And so if that key gets broken off in the ignition, so you can't get the key in there anymore, it stops your engine from working. And that's what these targeted drugs are doing. Now, the problem is that much like bacteria, when we use antibiotics, the bacteria are pretty clever. They find ways around these antibiotics. They find ways to grow without, in the presence of the antibiotic, if you like. And that's no different for the cancer cells. So what we know, for example, in adults that have been treated with these drugs that have ALK-positive lung cancer, the cells find a way to essentially reroute their circuitry. They get quite clever, they evolve, and they reroute their circuitry. So when we block this ALK protein, they don't care. They don't need it anymore because they find ways around it. And so a lot of work in my lab has been trying to understand what those rerouted circuitries are. How do the cells get around this protein that we've blocked so that they can grow without it? And this is work that Nina was doing in my lab. Nina was funded by a European Union project called Alcatraz and a project called Phantom Going Forward. And Nina did some really fantastic work looking at these resistance mechanisms. And one mechanism she was able to show was that what the cells do is normally downstream, they activate another protein called STAT3. And when you inhibit MPM-ALK, so it can't activate that protein anymore, the cells, as I said, they reroute their circuitry. They actually upregulate a different protein called the IL-10 receptor. And that protein sits in the membrane, but it does need an activator. But the cells get clever. They produce the activator themselves. And so they set up this circle where they become completely autonomous for growth. They don't need MPM-ALK anymore. They don't care if the inhibitor's present. They can find ways around it. And so the next step here would be to find ways where we can inhibit these other proteins that are downstream to prevent that from happening. Or if children should relapse with ALK inhibitors, to use this as a way to treat those children, to inhibit this bypass mechanism. So how are we testing these therapeutic approaches? Because of course we can't test them in the children for obvious reasons. And so we test them in mice. And it's not something we do lightly, but it's the best model we have, because of course many drugs need to be in the body to work and to be able to understand how they move around the body and how they interact with the cancer. And so we've managed to establish what we call patient-derived xenografts. So this is another way of saying we can take the tumor cells from the children and we can grow them on the back of the mouse. And when they've been growing on the back of the mouse, we can then try to cure the mouse of its tumor. And then perhaps we might be able to translate those findings to real patient benefit. And I'll just show you a couple of examples. So up here on the y-axis of the graph, this is the tumor volume. So what you can see here, if we just treat the mouse with the soluent, what we dissolve the drug in, you can see the pink line here, the tumor grows, it gets bigger. And you can see it here with the second patient, the tumor just gets bigger, and this is time. So within 3 days, this tumor is pretty much almost doubled in size. And then we can treat them with the different drugs. And so these are 2 different ALK inhibitors, crizotinib and brigatinib. And you can see through this patient on the right, when we treat that mouse with crizotinib, the tumor shrinks. It goes down to almost nothing. And when we treat the tumor with brigatinib, treat the mouse with brigatinib, the same thing happens. It almost shrinks it down to nothing. So we know that this particular tumor is sensitive. It will respond to these drugs. Now, the tumor on the left here, this one was grown in a mouse. And again, you can see that when we treat the tumor, the mouse with brigatinib, the tumor pretty much melts away and goes away. But this tumor, when we treated it with crizotinib, another ALK inhibitor, the tumor still grew. So it was very much resistant to this drug, but sensitive to this drug. And so this is just an example of how we can use this resource of what we call patient-derived xenografts, or PDXs, to mimic therapy, to essentially use it as an avatar to try and understand how treatments work. And we're also at the stage now where we can make these tumors become resistant to these drugs and use them to study why they're resistant. And so we started thinking about different ways we could administer the drugs that we have so that either resistance doesn't develop, or ways in which we can treat resistance once it's developed. And this is work that's being done by Lucy in my lab. Lucy is a clinical PhD fellow. She's training to be a pediatric oncologist and doing a PhD in my lab. And I'm going to tell you a little bit about these drug holidays that Lucy was working on. So we wondered what if you, instead of giving the drug continuously, which means that it's more likely the tumor will become resistant, what if instead we give it for a short spell of time and then give it a break and then come back and give the drug again in another block? Does that prevent the resistance, or at least delay it from happening? And I'm going to show you why we think that's the case. And so I'm going to explain what this is. So these are the tumor cells grown at this time in a Petri dish, essentially. And this is time along this axis here. So we put them in the dish, and then every 24 hours, pretty much, we look at how much they've grown. And this up here tells us how much they've grown. So you can see here, this is the tumor cells with no treatment, the blue line. So you can see they just grow. They grow really fast. But when we treat them with the ALK inhibitor, that's the red line, they don't grow. Okay. Now, what Lucy did was that she made these cells here, she made them become resistant to the ALK inhibitor. And she did this by growing them all the time, day in, day out, with the drug, and increasing its dose every so often, every couple of weeks, making the dose higher. And we got to the point where we'd managed to make tumor cells which didn't care that the drug was there anymore. They carried on growing. And what she did was to take those cells, the cells that are resistant with the ALK inhibitor, and she essentially could see that when she took them off the drug, so you can see here, there are two lines here that I want you to focus on, the green line and the pink line. And the pink line are the cells that are resistant to the drug, and we treat them with the drug, and you can see they still grow. And then what she did was to take the cells that were resistant to the ALK inhibitor and take them off the drug. So remove the drug completely. And then they stopped growing. So this was exactly the opposite of what we expected to see. When we took the cells that were resistant to the drug and we treated them with the drug, they carried on growing. This was a different, sorry, I've explained this completely wrong. So this is a different ALK inhibitor. So they're resistant to one ALK inhibitor, we come in with another one. But when we give them a holiday, the cells that are resistant to the drug, that's when they die. So essentially, they've become addicted to the drug. They need the drug to survive. And so by taking the drug away, the cells then die. So this gives us the basis for drug holidays. What if we treat them with the drug, the cells that are still sensitive will die, and that will leave the cells that have become resistant and have become addicted. Then we take the drug away, and those cells that have grown back will then die off. I don't think I've explained that very well. Apologies for that. And so that's what we're working with now, is looking at how we administer those different drugs and how we can essentially prevent this resistance from happening. I'm going to touch on very briefly now on ALK negative ALCL, which is exceptionally rare in children. And this is work that's being done by Emily, another very competent PhD student in my lab. And this is Rue, and I don't know if Rue's parents are joining us this evening, but Rue was one of those very rare children who had ALK negative ALCL and is now, I believe, doing very well, which is fantastic news. And Rue's parents set up a special named fund with the Children's Cancer and Leukemia Group and approached us and wanted us to look at the biology of this type of very rare cancer, because we know very, very little about it. And it's really hard to work on rare cancers. Fortunately, childhood cancers are rare anyway. But then when you want to work on a childhood cancer that's rare, you really are talking about very few cases. And so what we wanted to do to begin with is try and work out, because we see lots more of this cancer in adults, is ALK negative ALCL that we see in children the same as that we see in adults? And so we've collected 28 cases. Actually, I think we have a few more now from around the world, from countries everywhere, from Pakistan, Spain, America, everywhere. And we've analysed those cases. And what we found, I can tell you very briefly, is that they don't have any of these genetic changes that we see in the adult cases. So as I said at the beginning, we should never assume that a childhood cancer is the same as the adult equivalent diagnostic entity. It's very different. And so now what we're doing is we're doing what we call whole genome sequencing. So before, we've been looking at specific genetic events, so specific changes to the DNA code. Now we're scanning the whole of the DNA code of these samples to try and work out what changes in these tumors in children that makes them different. And that's work that is really underway now, and is work we're also doing with collaborators in Barcelona. I'm now going to move on to the B-cell lymphoma. Just check I still have some time. And so B-cell lymphoma, as I said, there are different types. There's Burkitt's, there's diffuse large B, there's primary mediastinal B-cell lymphoma, which is also very rare. And I'm going to focus on sort of Burkitt's and diffuse large B as one category together. And we've been doing a similar thing there. So this is Circa. Circa started working on this project in my lab with funding from the Alex Hume Foundation. And the Alex Hume Foundation, Alex, of course, was a young boy who unfortunately didn't survive his diagnosis of B-cell lymphoma. And I'm not sure if Dave and Nick or Matt, Alex's parents, and his brother are joining us this evening, but they've raised funding that's been enabled us to do the work that we've been doing today on Burkitt lymphoma and B-cell lymphoma in children. And so they funded Circa's PhD studentship. And part of what Circa did was to set up this collection of patient-derived xenografts of B-cell lymphoma. And she collected a number of cases from, again, all over the world, and we have them established in mice, which means we can now use those to really understand what drives this type of cancer. Sorry. And so this is where I move to my analogy to a beehive. And when we think of a tumor, when we think of a cancer, there are lots of cells that make up that cancer. They make up that mass that's growing. And we like to think that they're just all the same. They've all just stemmed from one cell and it just grew out of control. But actually what we know is that there are lots of different types of cancer cells. And they're all slightly different in their genetics, their DNA code. And this has probably occurred through an evolutionary process. So if you like, a survival of the fittest. So much like, evolution of people, the fittest survive. And so it's the same with the cancer cells. There will be more of the fitter population of cancer cells. So that's one idea. The other idea, which is a little bit more controversial, is that there is actually a stem cell. There's a mother cell. A cell that's there at the beginning, and that all of the daughter cells, if you like, come from that mother cell. And I prefer to call the mother cells queen bees. So much like a beehive, there are queen bees, and the queen bees produce the bulk of the tumor, and we call them the worker bees. But the worker bees, they don't do much, actually. They just sort of form the bulk of the tumor, but they don't really enable it to grow. Now, it could be the case that we have lots of different genetic subtypes of the queen bees, much like we do the worker bees. So they're not mutually exclusive models. So Circa set out to understand what's going on. And what she could do was she could take those tumor cells from patients, and she could find a small population of cells which drove growth of the tumor. And so these are the queen bees, the pink cells. And every time we put the pink cells into a mouse, they grew a tumor. So for every 3 mice that we injected tumor cells, all 3 of those mice developed cancer. But when we took the worker bees and put them into the mice, never did they grow a cancer. So this tells us that the queen bees are really driving cancer growth. And furthermore, what Circa could show was the queen bees, they're only about 1% of the tumor. So for every 100 tumor cells, only 1 of those is a queen bee that's driving tumor growth. So Circa went on to look at what these cells are. And what you can see straight away here is the queen bees, which are the blue line, if you can make that out. They grow. They always grow, and they grow really well. The worker bees, which are the black line down here, they never grow. And this is now in a Petri dish. So we started to wonder, how can you have a big tumor growing when only 1% of the cells are driving tumor growth? And B-cell lymphomas in children are really aggressive. They can double in size every 24 hours. I mean, not literally, but they get really big very quickly. So she did one experiment where she put the cells on their own or together and did different combinations. But one key part of this experiment was where she put the worker bees in one side of the Petri dish. And in the other side of the Petri dish, she put the queen bees. And she put a membrane between them so that they couldn't move between. But anything that these cells were producing, so any nutrients or growth factors, could move between the different sides of the membrane. And when she did that, she found that this worker bee population, they now start to grow. So what it seems is happening is that there is a queen bee population, but not only is that queen bee population producing the worker bees, they're also making growth factors or things that these cells need to survive and to grow. So it's like a double whammy. They're doing lots of different things. But another thing she was able to show is what we think might be able to happen is that some of these worker bees can become queen bees. And what we find is that no matter what ratios of queen bees or worker bees we put into a tumor, they always rebalance. They always equilibrate to find that 1% of queen bees and 99% of worker bees. So what is this telling us? Well, it's telling us that something is happening in this very small population of cells within the tumor. And so this is where Jamie, who's another PhD student funded by the Alex Hume Foundation, came working with us in the lab. And what Jamie helped Circa to do was to take those tumors in the mice, our xenografts, and the red lines show the times at which we treat the mouse. So this is the tumor growing. So when it gets to a certain size, we start to treat it with a chemotherapy protocol that's a bit like what we give to children with B-cell lymphoma. And you can see every time we treat the mouse, the tumor shrinks. And we could get it to the point where the tumor pretty much disappeared. But unfortunately, within a pretty much short period of time, that tumor grows back really quickly. So what is happening here and what is happening here? So when the first tumor is there and the relapsed tumor. And so Jamie and Circa set out to have a look at whether there were differences in the queen bee population. And sure enough, what they could find was that at relapse, that queen bee population had increased. So these are 5 different experiments. And you can see that that 1% of queen bees had become at least 5%, if not 8%. So there was a big increase in queen bees in the relapsed tumor. So what is this telling us? This is telling us we need to find out what's the Achilles heel of those queen bees. What drives those queen bees? Because if we can kill those, we may be able to prevent the growth of the big tumor. And what Circa and Jamie went on to do was to treat these queen bees with lots of different drugs. And a lot of these drugs you may know of, vincristine, doxorubicin, methotrexate, they don't affect those queen bees at all. They stay at 1% in the bulk tumor. She even tried some targeted agents, so some of those kinder drugs, so things like JQ1 and ibrutinib, sort of experimental drugs, and also established drugs, no effect. But she has managed to find one drug which does affect the growth of those queen bees. And I can't tell you what that drug is. It's an early experimental phase, so it's a long way off from being in the clinic, but it does seem to affect those queen bees. And this is work which is now moving forward, still with funding from the Alex Hume Foundation, but also from the Little Princess Trust. And where we are now clinically is that we are now working on a new clinical trial, which will hopefully open towards the end of this year. And this is a trial that is being led by a very good colleague of mine here at Addenbrooke's Hospital, Amos Burke, who is a pediatric oncologist, who not only works on the ward and treats children, but also dedicates a lot of time to helping us in the lab. And his input is absolutely pivotal to us being able to understand what the problems are in the clinic so we can design our research so that we address those problems. And Amos is the chief investigator for a very new trial, very novel trial that's called GLOBE-NHL. As the name suggests, it's a global trial. It's going to recruit children all over the world. And these children are children that have relapsed with B-cell lymphoma. So fortunately, there aren't many children that relapse, hence it's a global trial. But for those children, we need to be able to do something. And we're going to be treating those children with a new class of drugs, if you like, called immunotherapies. So drugs which harness our own immune systems to try and do something to help these children for whom, at the moment, we have very little we can do to help them. And the biological studies which are underlining this trial are funded by the Little Princess Trust. The trial itself is funded by Kick Cancer and also Cancer Research UK, and is being run from the Clinical Trials Unit in Birmingham. And the GlobeIN8 trial, and I have to add, of course, the Alex Hume Foundation are also funding the biological studies in this trial. What this trial will do, as I said, is recruit those children and give immunotherapies. And I'm just going to touch briefly on what immunotherapies are, just to try and help you understand what they do. And there are different types of immunotherapies we're going to use in this trial. There are things called CAR T cells, which you may have heard of in the press. There's been some great successes with CAR T cells. There are things called antibody drug conjugates, which are essentially antibodies which have been generated against the cancer cells. And then there are things called BiTEs, or bispecific T-cell engagers. And basically what they are is CAR T cells. We harvest T cells from the child, and then we send those T cells to school. So we send those T cells to school so that they learn to recognize the cancer cells. And so then when we put them back in the child, they will circulate in the body and hopefully will target those cancer cells and essentially eat them up and get rid of them. That's the hope. And we train them by giving them a little flag on their surface which recognizes the cancer cells. And that's what the CAR part of it is. It's the little flag which recognizes the cancer cells. Then there's the antibody drug conjugates, which are essentially antibodies. And there are a couple of different ways that we can use antibodies to recognize cancer. So some cancer cells have flags on their surface. And if we think about B-cell lymphoma, so Burkitt's lymphoma, some diffuse large B-cell lymphomas, they express a protein called CD20. Now, this is a little flag on the surface. And what we do is we have an antibody that recognizes CD20, and this is called rituximab, which you may have heard of. And hopefully many children with B-cell lymphoma are now being treated with rituximab as standard care with the standard chemotherapy. And then what happens is that these cells are essentially flagged up to the immune system. Now that this antibody rituximab has bound to them, the immune system now recognizes them and gets rid of them, and comes along and eats them up. And there's another type of antibody therapy that can be used, and this is more of the sort of cytotoxic form of therapy. Now, some cancer cells express a protein called CD30, and this is the case for anaplastic large cell lymphoma, the T-cell lymphoma I was talking about in the first half of my talk. And what we have here is a drug called brentuximab. And brentuximab is a little bit different. What brentuximab does is it has a little bomb attached to it, essentially. This is a toxic protein, a toxic molecule. And this whole thing then gets internalized into the cell, at which point the bomb is let off. And so that's another way we can target these cancer cells, is by using this sort of approach. And then there are the bispecific T-cell engagers, or the BiTEs. And in very simplistic terms, what they do is they form a bridge. So if we have a tumor cell, for example, and it's a tumor cell that expresses CD20, the bispecific T-cell engager will recognize that flag. And at the other end of the bispecific T-cell engager, or the BiTE, it will recognize a flag on the normal healthy cells. So in this case, this is recognizing a flag called CD3, which sits on normal healthy T cells, and it brings them together. And the hope is that by bringing them together, you encourage the normal healthy immune cells to kill the tumor cells. And so these immunotherapy approaches are going to be the basis of the GlobeIN8 trial, which, as I said, we hope to be able to open towards the end of this year. And so that we have something that we can offer children that have relapsed with B-cell lymphoma. So take-home messages, what can I tell you? So I've realized I've just garbled through a whole lot of information. And really, just I want to impress on you that we really are working very hard in the labs. There's a lot of research being conducted to try and improve the lives of children with lymphoma. They're not forgotten. Survival rates for some forms of lymphoma are pretty good. And so many funding agencies don't see there as being a problem there. But fortunately, there are charities like the CCLG, the Little Princess Trust, the Alex Hume Foundation, who recognise that we really need to be able to do more. And with their support, we are able to do that. And so we are developing kind of treatments that act specifically on the tumour cells, or treatments that encourage the immune system to kill the tumour cells. That work is all going on, and it's all moving ahead into very early clinical trials. But research is a team effort. And by a team effort, I don't just mean us in the lab and the clinicians on the ward. I also mean the parents, the family, the children with cancer. We really need your support and your input into our research, because without that, we don't fully understand the issues that you face with your children undergoing the therapy that they are. So please do get involved. Please do get involved through the CCLG, through the charities that support the work that's being done on the whole paediatric oncology community. And I'm going to end there with some acknowledgements, because it's very important that I show appreciation for the people that really do this work in the lab. So I'm the person that really sits behind the computer and makes sure the people in the lab are able to do what they need to do. And these are the current members of my lab here. And some of the members of my lab work on B-cell lymphoma, so the guys at the top end here. And some of them, such as Martin and Benjamin, work on endemic Burkitt lymphoma in sub-Saharan Africa. And that's work funded by Global Challenges Research Fund, Albarado Trust, and also Great Ormond Street Charity, as well as the Little Princess Trust. Some members of my lab, such as Lucy, Emily, Carla, Man, Hannah, work on anaplastic large cell lymphoma, so the T-cell lymphoma, and that's work that we've been conducting with funding from the European Union through the Phantom and Alcatraz networks, also through CCLG special name funds such as Superru. And I know that there are a number of special name funds that have been set up now by the parents of Lila, Annika, Neve and Elliot, who I really hope that we can work with going forward to make sure that the therapies we have available are going to work and really lead to improved outcomes. Of course, I should have also mentioned Francesca's Research Fund, who have also been supporting the B-cell work, and of course, Little Princess Trust too. And then I have members of my lab, Perla and Charlotte, who work on neuroblastoma, which is a different form of childhood cancer. And we're all supported in the lab by Layla, Rohir and Jill. And of course, a multitude of collaborators. When you work on childhood cancers, which are fortunately, as I said, rare, you do have to work with people all over the world, which is fantastic that we are able to come together to approach the problems that are present and really find solutions. And I'm indebted to my collaborators who've really helped us get to the stage we're at. And I'm very happy to take questions.
Okay, so thank you very much for that talk. It's really interesting and very engaging. So we do already have a few questions. We are running over a little bit, but the recording will be sent around if anyone has to log off. So I think we'll start with this question from Bev, if that's okay. So her son had, I think it's diffuse large B-cell lymphoma, DLBCL.
Yes.
Okay. Yes. Good. Yay, acronyms. And in 2018, and then grew a new tumour that wasn't the same thing in 2021. She wants to know if you are looking at genetics and immune disorders as something that triggers lymphoma, as he grew 2 lymphomas in 3 years.
Yes, so there are, and of course I can't comment on your specific case. I'm not a clinician, so I just put a disclaimer out there. But there are forms of lymphoma that occur in children, which occur in children that do have immunodeficiencies, so their immune systems don't function in the way they should. And this is a very small subset of lymphomas that we see, but they do exist. There is work going on to try and understand those at a European level, at least, well, actually an international level. And we've actually just set up a new working group. And we're going to try and collect as many of these cases as we can so we can try and understand a bit more about them. But it's really a global project. It's going to take some time to get enough cases so that we can get some really, really meaningful data out of it. But yes, there is work that's going on to look at predisposition to lymphoma. So, and some of that is due to children who are slightly immunodeficient or have a sort of inborn immunodeficiency. Does that answer your question? I'm very sorry to hear that your child has developed a second cancer. I'm sure that's been very difficult.
So I think our next question is from Wenjun, who wants to know, for relapsed and refractory ALCL, what's the best treatment and medicine? Can weekly vinblastine injection cure it? And I know, obviously, as you say, you're not a clinician. We do have a few questions about specific situations.
Yeah.
But
so relapsed ALCL is fortunately one of those lymphomas which it doesn't seem to matter what we throw at it, that children tend to do very well. So for some children, single agent vinblastine, so just the one drug given weekly probably for 2 years, so it's quite a lengthy treatment, could potentially cure them. And there is a trial for children that is currently running in Germany, where children that have the really good antibody responses and don't have disseminated disease, if we give them weekly vinblastine for 2 years, we're hoping that will be enough to treat them. But on relapse, it's slightly more difficult. We don't understand a huge amount about ALCL that's relapsed from standard treatment. But what we do know is that generally we can rescue children that have relapsed with ALCL, and single agent vinblastine is one of those approaches. Another approach is to get to the point where a transplant is possible. So a bone marrow transplant, put back some nice healthy cells. And that's another thing that can be done for relapsed ALCL, but it varies very much on an individual basis, and your clinician is best placed to advise you on that.
So our next one is also about ALCL. So Philip would like to know, can we use CAR T therapy to treat ALCL? As you mentioned, there's a trial being run that's for B-cell lymphomas, and obviously it's a different type of lymphoma.
Yeah. So CAR T cells rely on there being a flag on the surface of the tumor cells that's different from what we see on other cells. And for ALCL, that flag is CD30. So if we could develop CAR T cells to CD30, then yes, it's quite possible CAR T cells could be used. I'm not aware of anybody doing that at the moment. That's not to say it's not going on, but yeah, it's entirely possible that CAR T cells could be used at some point. And again, that's very much varies from child to child, because you have to be able to harvest healthy T cells from the child to be able to start making those CAR Ts and for that sort of therapy. So again, it really depends on each particular child and what their status is.
Okay, so next one, and this is one I was curious about actually as well, because obviously the drug holidays idea is very different to antibiotics and antibiotic resistance. So Abraham would like to know about the drug holidays. Don't we expect the addicted cancer cells to grow again when we restart the patient on the drug, or do we go for a better drug after the holiday?
Yeah, it's going to be a very tricky thing to do. And we need to, before we can implement that sort of thing, we need to find ways of new biomarkers to be able to monitor the exact situation that the tumor is in. Because as you say, you could get to the point, whereas if you have these two population of cells, you never know at any one point in time exactly how many of each you've got, and it'll be very much a balance. But having said that, I know of at least 2 children, not in this country, but children who've been treated with ALK inhibitors on that basis, who are doing very well. And what they do there is they monitor the presence of those tumor cells in the blood. So they give the drug for a week, and then they wait for a week, and then they have a look and see if there's any of that disseminated disease. So can they pick up any evidence of those tumor cells in the blood? And then make a decision, do we come back in again or not? And actually, they have seen really nice responses. But this isn't in a trial setting. This is a one-off case, and of course we don't have controls there, so we don't know if that child had been treated continuously with the drug, whether they would have had the same response. So as with all new cancer drugs and new ways of treating children, it really has to be done in a trial setting so that we can fully understand the implications of these approaches.
Thank you. So we've got a question from Jana. I'm not sure if you're going to be able to help much on this because it's a bit more specific, but I'm going to ask it you anyway and give it a go. So she says her son is ALK negative and chemo didn't work. Now he's trying nivolumab. Mm-hmm. Do you know of any other treatment options? And she says that they are based in Germany.
Yeah, so yeah, I'm sorry to hear about that. So ALK positive LCL, as I said, is extremely rare, and so we treat children with. Sorry, did you say ALK
negative?
Yes, ALK negative. Yeah. Sorry. So we treat children with ALK negative ALCL the same way we do with ALK positive ALCL. So I'm sorry to hear that that hasn't worked. Nivolumab is being used in a trial setting, and hopefully that trial will open in the UK also this year. Using nivolumab, as you said, and nivolumab is one of those forms of immunotherapy. So what it does is essentially it switches off the signal which tells T cells not to eat the tumor cells. So it's like a switching off of the don't eat me signal. And so the tumor cells sort of become alive to the immune system again, and the immune system kills them. And there has been a lot of really nice responses with nivolumab in children that have relapsed on the standard therapy. And remember, of course, even the ALK positive children, for the most part, they're not receiving ALK inhibitors at the moment. Most of them are getting the same protocol, which you may know as being ALCL 99. That's the name of the therapy protocol they get. And those children also go on to nivolumab when they relapse and again have had very good outcomes. So hopefully nivolumab will do the job and there will be a really good outcome for your son with that. And then I guess going forward, we hope that there will be no need to approach anything differently. But I would suggest you speak with your clinician, and there are some very well versed clinicians in Germany who have done some fantastic work on the ALK antibodies and looking at treating ALK positive ALCL and ALK negative ALCL based in Germany. So I'd encourage you to speak with your clinicians some more, but nivolumab is a very good approach.
Okay, so I do have another specific one. So Trisha's daughter has just relapsed with a skin lesion that's ALK positive. They're currently working out treatment plans and everything. She has so many questions, but the one that she's put in here is, how do you test to see if they have ALK antibodies?
Yes, so it's a test we do in the lab. It's not a test that's available at the moment, and that's because it needs to be validated in a trial setting first. So at the moment it's very much an experimental technique. We do it in my lab here in Cambridge, but it's not been validated as a clinically actionable test yet. So it's very much experimental. And so yes, it can be done here in Cambridge, but we don't really understand how we can use it in the clinic just yet. So it's hopefully something that will come about to be more widely available in the coming couple of years.
So is it like a blood test that you're sort of running on, almost COVID test kind of thing? So
yeah, exactly. It's exactly like a COVID test. So you take some blood, and you essentially put it onto a disc like you would for COVID, and it's a bit longer. It's about a 3-hour test. It's a little bit longer than the COVID test, and then you look for a positive signal, and we can measure how positive that signal is. It tells us how many antibodies are there. But the assay itself has not been developed by a company. It's been developed in a lab. It was actually developed by a group in Oxford initially, and it's a very difficult assay to do, and so we need to do a lot of work to get it to the point where it's an assay that can be used in hospitals very quickly and very easily.
Okay, so the last question is another one from Philip. So he would like to know, is there potential to use CRISPR gene editing to treat ALK positive ALCL cells that have become resistant to the inhibitors, and why not use CRISPR to make the resistant cells sensitive to the LNK inhibitors again?
Yes, CRISPR is really quite a powerful technique, and at the moment I'm pretty sure CRISPR isn't approved for any human applications because we don't really understand enough about its power. And you may have heard things in the press about CRISPR being used to make designer babies and this sort of thing because it is so powerful. We use it as a tool in the lab. But I think we are quite a long way off from it being used for any sort of therapeutic approach. But yes, ideally that would be one approach you could take, would be to modify the cancer cells with CRISPR, but you would have to do it outside the body. I can't think of a way that it would be easily implemented yet, but certainly it has a lot of potential.
So I'm just going to sneak in one of my questions just to finish us off because I didn't get a chance to ask any. So just one thing, you talked about the bypass mechanisms when they decide that they don't actually need to use the other one anymore, they use other ones. Are the mechanisms inbuilt to all cells, or is it specific, like due to cancer mutations, or what is it? I guess is the question.
It doesn't seem to be driven by mutations. It just seems to be rewiring of what's called the transcriptome. So that's how the DNA is read, the bit that's the next level up before you get a protein. And we really don't fully understand how that happens. But what we do know is that it will likely vary from patient to patient. And so we will have to be able to get tumor cells at relapse, so a new biopsy at relapse, so that we can then look at what's changed about the transcriptome in those relapsed tissues. And that's something we're working with. So we're also working with the ALK-positive lung cancer because it will be slightly easier. There are more adults with ALK-positive lung cancer than there are children, but also bringing in the data we have from children. And we're also working with astrophysicists of all people, and people who do some really clever stuff with computers so that we can try and predict what might be happening in the patient's tumors from what we know about what we see in the lab. So that's something we really hope to take forward. And my ideal situation for me, my goal would be, I get to the situation where we have a test where we can take some blood from a child and we find remnants of the tumor in the blood, the DNA or the transcriptome from that child's tumor, and we run its sequence through a machine, and what pops out the other end is the drug we need to use. And so that's where we want to be. And we're slowly making our way there. It's a lot of work, and it's getting quite complicated stuff. I have to say a lot of it is straight over my head. And but hopefully that's where we will get to at some point in the not too distant future.
Oh, it sounds fantastic. So fingers crossed.
Yeah,
I know it's down to a bit more than luck, but I'll keep them crossed anyway. So I think that is the end of all our questions. So I will say a little bit at the end. So thank you all very much for joining us this evening, and thank you to Suzanne for your talk and for all of your insightful answers.